Very good. Okay, so what Lydia will talk about a little bit after me is about controlling cell signaling. So I will give a little intro about why we do this and what it is for. Every single time we look at cell signaling in books, or this is actually an advertisement from actually cell signaling, which is a company that sells antibodies, and we see a pattern of receptors that usually are activated and bring other proteins to their vicinity. They form some complexes. Then they trigger a bunch of cascades of downstream signalings in signaling pathways, which involve usually kinases activated some other kinase, activates another kinase, and so on, forming later on protein complexes and sometimes protein RNA complexes that translocate from one place to another. And this often triggers cellular responses like transcription, translation, or metabolic changes. And among these things, we also see that there is some molecules which are always in between. Yeah, I'll show you another example. This one, for example. Yeah, so all pathways just seem to go through this to some of these molecules. Now the question is how cells figure out, what is what, because all no matter what receptor you activate, you actually turn some of these kinases on, for example. But cells do know which signaling pathways are activated. So the question we've been asking for some time is if we are able to have like a single switch, or every single time we will have to go through a series of switches to get a proper signal. And for that, we turn into light. And the reason we turn to light, as you can see on this video, is because light is something you can control with incredible precision. You can point at things with light, and you can control its timing. And of course, it comes in a variety of colors. Now, why light? Well, first, because organisms, they already have proteins that react to light. Yeah, obviously, the whole photosynthesis, and of course, all the other creatures that love to go to, whatever is nice and warm places. And we already have proteins that sense light, like the opsin receptors in our eyes. That's how we actually see. We see photons through the detection via these cells that they have these receptors. Now, the origin of light-induced research, it comes from a slightly different set of receptors, which are not the opsin receptors as such, but they are kind of from the same family. Unlike our receptors, the opsin receptors, the ones from microbes, they are not G protein coupled receptors. So they're actually channels. So like the channel rhodopsin. And these channels just essentially open and close when they are exposed to light. And by adopting these channels, mostly in neurobiology, they have been able to regulate the way that neurons turn on and off, by polarization of neurons. And as you see, there is an image from a few years ago where they were starting to control different neural circuits using light. Yeah, they have these transgenic animals that they express these receptors in a particular set of cells. And they have managed to create false memories and even allow making animals to forget things, just like these guys did. For example, they have mice that they made them to forget literally that they are afraid of cats. They didn't put the mice with the cats, but they actually just put a smell of cats, which is enough to make mice run to the corner of the cage. Anyway, these very interesting experiments, they trigger the search for many, many other molecules. Many of them are not receptors anymore, which also react to light. And as it happens in science, they went to look for them in all kind of organisms, mostly plants, not surprisingly, because they have molecules that obviously respond to light. And many of them are dimerizers, so that means two molecules get together, two of the same molecules, or sometimes two different molecules that get together when they get activated by light. Some are actuators, some are channels, as I mentioned before, and so on. And what Lidia will explain you in a minute is how we use some of these systems to control a very particular signaling pathway. So I stop here and I leave the chair for Lidia.
So as Adolek introduced me, I'm a postdoc in his lab in Department of Biochemistry and Molecular Biology at Medical University of Lublin. I would like to thank for this opportunity to give a talk about our work. It's about controlling notch signaling. So the first data about notch signaling comes from 1914 when John Dexter described characteristic notches on the wings of Drosophila, and then a few years later Thomas Hunt Morgan identified the notch gene, and basically the notch it's a receptor protein which is anchored to the membrane and activated upon binding to its ligand expressed on a signal sending cell, and then activated notch triggers downstream responses. So there are 4 notch receptors, notch 1, 2, 3, and 4, and they possess as you can see, the extracellular domain, the transmembrane domain, and intracellular domain. And also there are 5 different notch ligands belonging to jagged serrate family protein, jagged 1 and 2, and to delta-like type family protein, DLL 1, 3, and 4. And this pathway, this signaling pathway, is important for determination of cell fate. And here we've got like 2 examples of the possible outcome of notch activation. So lateral induction happens when notch activated by binding to the ligand triggers the expression of the ligand on the surface of the cell, and the signal receiving cell is becoming also signal sending cell. So the signal is transmitted to longer distances, not just to neighboring cells. And the opposite outcome is lateral inhibition, when the notch activated upon ligand binding is inhibiting expression of the ligand on the signal receiving cell, and the signal is not transmitted to other neighboring cells. And this happens, for example, during the vasculature formation, when in the tip cell there is overexpression of DLL4 notch ligand, which of course is binded by notch protein in so-called stalk cells. But then notch is triggering the expression of jagged protein, and jagged ligand is expressed on the signal receiving cell and send the signal to further. So we've got at one end the suppression of the tip cell phenotype, But then we've got also the induction of the stalk cell phenotype at the molecular level. So upon binding its ligand, notch is activated, and this triggers like a series of proteolytic events. So first, the ADAM metalloprotease family protein cuts the extracellular domain. Then it reveals the cleavage site for gamma secretase protease. And after that cleavage, NICD is released from the cell membrane. And the intracellular domain of notch, NICD, translocates to the nucleus, where it binds to MAML and other transcriptional activators to activate the expression of its target's genes. And this scenario happens like whatever, no matter what ligand binds to notch. So these events will be the same in terms that notch will be cleaved and the NICD domain will translocate to the nucleus. So it was like very intriguing, like how cells distinguish between different ligands to trigger specific response to the signal. And this caused a need like to develop some model to study notch signaling in a cell. And there were several such models. First, the first approaches was to attach ligands to the inert surfaces. Then, thanks to bioprinting and microarray, we got like the some kind of functional surfaces expressing notch ligands. Other approaches were substituting the ligand with GFP, here presented as a green barrel, and the extracellular domain was exchanged with the nanobodies recognizing GFP fused with NICD, which were then translocated after cleavage to the nucleus. And these attempts were really helpful to study how the number of ligands, their aggregation, their density, their agglomeration on the surface, how these factors can modulate notch signaling. And also there were several optogenetic models to study notch signaling. So first from the left, it's the optodelta system, where the delta ligand was fused with Cry2 photoactivable protein. And this protein aggregates under blue light stimulation. So after blue light stimulation, we've got an agglomeration of the DLL ligand on the surface of signal sending cell. And as I showed you before, the overexpression of DLL causes the signal inhibition. Also, there were some optogenetic models that were independent from the ligand. For example, Tango Light system, which is composed of the membrane anchored chimera of the photoactivable CXCR4 receptor fused with the C-terminal of vasopressin receptor containing also a TEF protease recognition site, and of course NICD. And upon activation, this CXCR4 photoreceptor recruits beta-arrestin and TEF protease to the complex, what results in a proteolytical cleavage and release of NICD from the plasma membrane. Also, optic notch system in the middle here, which is anchored to the cell membrane with two photoactivable proteins, CYP1 and LOF2, which in the dark are masking the TEF recognition site. And also they were fused with one half of TEF protease. And in this, in the dark, this complex cannot bind with Cry2 complementing half of TEF fusion protein. So the NICD stays anchored to the membrane. But upon under blue light, like two phenomena occurs. First is Cry2 and CYP form a heterodimer and reconstitute the TEF protease. And the second is unfolding of LOF protein, which uncovers the TEF protease recognition site. And this results, again, in proteolytical release of NICD from the membrane. And the two last systems are very similar, opto-notch and opto-NICD. They are similar in their structure because they consist NICD fused to one partner of the heterodimeric photoreceptor protein LOF2's dark1, which dissociate under blue light stimulation. And in opto-notch LOF2 protein, here it's a green triangle, is anchoring complex to the cell membrane, while in opto-NICD, the ZDK protein here marked as a magenta diamonds is anchored to mitochondrial membrane. But upon light irradiation, the NICD is released from both of the system and goes and is translocated to the nucleus. And two of the presented here systems were developed in our laboratory. It's Tangolite and the opto-notch system. And today I would like to tell a bit more about the opto-notch system. So what we wanted to achieve is we want to have a system, a model that allows to control notch signaling with spatial and temporal precision and independent of the ligand and gamma secretase. So we designed a system where in human cells, NICD is anchored to the membrane together with LOF2 and dark1 proteins, which, as I told you before, form in a dark heterodimer. But they dissociate after blue light stimulation, what results in release of NICD, its translocation to nucleus, and expression and triggering expression of notch target genes. So for the light stimulation, we are using a platform with LED lights, which we can put into the incubator with the cells. So we are avoiding the changes in environmental conditions during light illumination. And we were optimizing like the pattern of illuminating cells with the blue light. Here I'm showing just the one which was the best which worked the best for our experiments. So it was 5 milliseconds of blue light pulse and then break for 5 seconds and again 5 milliseconds blue light pulse. So first we wanted to check the performance of our system. and we transfected the cells with opto notch, and we will monitor the localization in the cell, the localization of the NICD with the immunostaining. Here at this picture, we've got the cells labeled with hexed. The nuclei are in blue, and as you can see, the NICD is expressed in the cell membrane. Here we see the green signal in a whole, equally distributed in a whole cell. This is situation in the dark. And under blue light, we observed that indeed the NICD is released and it translocates to the nuclei. So this was like a first proof that our system works. Then we wanted to optimize the conditions for its performance. So we transfected the cells with optonotch, and at the same time with known CSL luciferase reporter systems, which enables to measure the luminescence of the luciferase, which corresponds to activity of optonotch stimulation. And as you can see on the top right, upon light activation, we could see a significant increase of the luminescence in the cells. And we also checked the notch target genes expression by qPCR, and we see that known notch target genes like HES, HEY, and Notch3, which are known that are upregulated upon Notch1 activation, were exhibiting significant increase in expression after blue light stimulation. The notch signaling, as I showed you before, is important for the angiogenesis and vasculature formation, and also it's important for embryonic development, cell differentiation, and homeostasis. But the regulation of this signaling pathway has a serious outcome, and it causes development of number of diseases, including cancers. And in cancerogenesis, it's important for the metastatic and stemness, and also chemoresistance of the cancer cells. So we decided to test the performance of our system in a breast cancer model. I'm showing you just how we planned the experiments. So we transfected the breast cancer cells with the optonotch, and then we light activated some cells. Controls were not activated, and also we are observing as a negative control the not transfected cells. The notch signaling is reported to play a big role in proliferation and migration of breast cancer cells. So we did the scratch test experiments to check if what is the effect of optonotch on proliferation and migration of two breast cancer cell lines. For this experiment, we choose the MCF7 estrogen positive breast cancer cell line, and the triple negative, more aggressive MDA-MB 468 cell line. And also for these experiments to inhibit the endogenous notch signaling, we were using DAPT chemical, which is inhibitor of notch signaling. In particular, it inhibits the gamma secretase. So as you can see in both cell lines, after light irradiation, we observed the increase in proliferation and migration of the cells. And also to confirm the influence of notch in breast cancer development under physiological mimicking conditions, we cultured optonotch expressing cells in 3D spheroids in matrigel matrix, and we observed that under blue light stimulation, the size of spheroids increased faster in the optonotch transfected cells than in this not illuminated with blue light. We also took a look on the morphology of spheroids, and we observed that upon blue light stimulation, the MDA MB468 cell line exhibited more aggressive phenotype, but cells were less likely to migrate out of the spheroids. And in the case of MCF7, we observed a more invasive phenotype. This also reflects the different characteristics between these two breast cancer cell lines. And since the increased activity of notch was reported to induce resistance of breast cancer cells to drugs, we decided to analyze the chemoresistance of the optonotch transfected cancer cell lines. And for this experiment, we chosen two drugs, the doxorubicin and paclitaxel. And what we observed that light activation induced doxorubicin resistance in MDA MB468 cells and paclitaxel resistance in the MCF7 optonotch cells, significantly increasing the proliferation of the cells. And here, just to summarize, the take-home message that notch signaling plays a multi-phase role in cell development, communication, and differentiation via receiving extracellular signals and transmitting them from neighboring cells to the nucleus and triggering the expression of its target genes. And the optonotch system allows us the millisecond control of notch signaling, both independently of cell-cell interaction or gamma secretase activity. Until today, it's only available approach for studying the strength, frequency, and pattern of the notch activation by modulation of light irradiation intensity, as well as number of frequency of the light pulses over time. So by modulating the light intensity, we can trigger the specific response in the cells. And in the future, we would like to also move to in vivo model and to create transgenic zebrafish with the optonotch system to control notch signaling in vasculature. So this system is very plastic, and we hope to move from in vivo studies to in vitro. And here it's a presentation of our team in ARMS Lab. Here also I would like to acknowledge Joanna Kałafut for her great effort in studying optonotch. And thank you for your attention, and I'm happy to answer your questions.
I might have a question. Great presentation, thank you. I was wondering how dynamic is, like, it's a very general regarding, I don't know if the system actually I know more about the system. So actually I have two questions. So one, if you want to use the kind of question, do you have to use CRISPR or like, is it like just a transfection? Or you have to have like a stable introduction of the fusion protein to your cells. And then how dynamic is if you can you like modulate, is it like on and off signal, or you can kind of like dose it? So for instance, with a certain light wavelength, you have like a little bit of the effect or like just shorter effect, or then if you increase it or prolong it, it's gonna, like for instance, dimerization will have it for a longer time, or in yourself, I understand that it's fusion, but if it's gonna be for instance dimerization, then it's not like modulate that.
Yeah, so regarding this first issue, you can achieve that with like transient expression, but then you're limited in time, like in performing your experiments. So yeah, most of these experiments were done in stable lines, expressing constantly up to notch, like nothing happens. It's not affect the morphology of the cells, and they are acting the same as a wild type. We were testing it in different cell lines, in cancer, but also in normal cell lines, so it is okay. And it's possible to obtain stable cell lines, then you've got no problem with the efficiency of transfection all the time. And regarding like the second issue, yeah, you can play with the light. And then you've with the intensity of the light, and with the timing, like how long and how many pulses. And this is also like the characteristic of the this happens also in the nature. I mean that to activate notch, DLL needs like shorter contact, shorter binding, and it activates notch. While for the jagged one, it's like longer contact, and then we've got a boom. So yeah, this is a great feature of this system, that you can play with light, which is like, you know, easy, quite easy, much easier than playing with the density of the ligands, or keeping at contact ligand and notch. Yeah, so this is the big advantage of this system.
Okay, so if we don't have any more questions, you can always text or email Lidia with more questions, or myself. We're happy to answer them. And one thing that Lidia forgot to mention is that actually notch signaling is involved not only in transcription, but in translation too, and in several types of cancers. So it is in a way connected with Translacore, because downstream some notch signaling, there is several ribosomal proteins. And we know that from some previous experiments with knockout cells and stuff like that. So yeah, so here is the connection between Translacore and notch too. Right, Lidia?
Yeah, so we can fix it on YouTube.
No, we can fix it on YouTube. Yeah, we add it later. Yeah. All right, something else?
Just mechanofixing, yes, on YouTube.
Mechanofixing, yeah, exactly. Okay, if not, then I will actually stop recording.